Bismillahirrahmanirrahim. Quote for today is: It is never too late to be who you want to have been. Today, I will be doing the critical appraisal of an article that is already published under the supervision of my professor, Dr. Saira Afzal. The article that I am going to critically appraise is folate deficiency reduces the development of colorectal cancer in rats. I will be using a RIFE checklist for this article to critically appraise because this is an animal study, so this list will be used. This slide shows a RIFE guideline checklists. There are 20 main points. I have attached screenshots of the list. One is this and another is this. Now the title of article is Folate Deficiency Reduces the Development of Colorectal Cancer in Rats. While title according to a RIFE checklist, it says that it should be provide as accurate and concise a description of the content of article as possible. This step was not followed because in the article it is explained that folate deficiency reduces the azoxymethane-induced intestinal tumors. While according to title, it is the title shows that colorectal cancer that is caused by any reason is reduced by folate deficiency. So according to me, this title is not very good and it's not concise. Abstract of the article is: the aim of the study was to examine the effect of the diminished folate status of azoxymethane-induced intestinal tumor in Sprague Duvalier rats. A total of 125 weanling male rats were divided into 5 equal groups and fed semi-purified diets containing either 8 mg per kg folate or no folate. After 4 weeks on experimental diets, all animals received 3 weekly subcutaneous injection of azoxymethane at a dose rate of 15 mg per kilogram body weight. The animals were necropsied after 26 weeks. Rats with a diminished folate status, evident by the significantly reduced blood levels and colonic folate concentration and elevated plasma homocysteine levels, had significantly lower incidence and number of small intestinal and colonic tumors compared with rats displaying adequate folate status. Now, I have highlighted this portion that there was a significant decrease in the incidence of colonic adenocarcinomas and size of colonic tumors observed in the rats displaying a diminished folate status. This study shows that a diminished folate status was associated with the decrease in the development of azoxymethane-induced colorectal cancer. The decrease in risk may be attributed to the known role of folate in cell multiplication. Now, abstract according to ARRIVE checklist, that it should provide an accurate summary of the background, research objectives, includes details of the species or strains of the animals used, key methods, principal findings, and conclusion of the study. While according to this checklist, background is not given in the abstract. Aim of the study, however, is given. Details of the species are given. A key method is not given. Principal findings and conclusions of the study were also given. I've added only 2 paragraphs from introduction that were important. Dietary factors are thought to play a predominant role in the causation of colorectal cancer. Diets high in fat or meat and low in fruit, cereals, or vegetables have been found to be associated with a higher risk of colorectal cancer. The reduced risk of colorectal cancer in association with consumption of fruits, cereals, and vegetables may be explained by certain micronutrients. A higher intake of the micronutrient folate was first proposed by Frudenham to reduce the risk of colorectal cancer. Folate deficiency is one of the most prevalent vitamin deficiencies worldwide since the case control study conducted by Frudenham. Other epidemiological studies have tended to suggest an association between diminished folate intake and increased risk of developing colorectal cancer. However, none of these studies relate actual folate deficiency with increased colorectal cancer risk. The purpose of the study was to determine whether a diminished folate status might alter the development of intestinal tumors induced by azoxymethane in male rats. A secondary objective was to test if DNA methylation was altered in the folate depleted groups. A non-absorbable antibiotic drug was employed in same dietary treatment to further enhance the level of folate depletion both before cancer initiation and during the promotional progression phase of carcinogenesis. Now, introduction according to ARRIVE checklist. According to ARRIVE checklist, background should include sufficient scientific background, including relevant references to previous work, to understand the motivation and context for study and explain the experimental approach and rationale. This point was followed in the introduction. And next point was explain how and why the animal species and models being used can address the scientific objectives and where appropriate the study's relevance to human biology. This point was not followed throughout the introduction. Next is objectives. It should be clearly describes any primary and secondary objective of the study. Primary and secondary objectives were told in the introduction, and the specific hypothesis being tested, there was no hypothesis given. However, with the heading of objective, this thing was not given in the introduction. It was just said that the purpose of the study and the aim of the study is this. Methods. A total of 125 weanling male rats that were of weight 70 gram were purchased from the Animal Resource Center in Australia. Animals were housed in stainless steel wire bottom cages maintained in air-conditioned environment with a 12 hours light and 12 hours dark cycle. At the age of 4 weeks, they were sorted into equal body weights and divided into 5 dietary treatment groups of 25 animals. The experimental protocol and the use of rats was approved by the CSIRO, that is the Health Science and Nutrition Animal Experimentation and Ethics Committee, before commencing the study. Next in the method is sample collection and autopsy. Fresh fecal samples were collected from each rats after 4 weeks on experimental diets. Pieces of the known weight were placed in 3 ml deionized water and homogenized. pH was read and then the samples were stored. The rats were killed by exsanguination while under halothen anesthesia after 26 weeks on experimental diet. Blood was collected into EDTA-treated tubes. The animals were fed into experimental powdered diets and given feces free access to distilled water for a period of 26 weeks. Body weights were recorded weekly. After 4 weeks on experimental diet, animals were injected subcutaneously with the drug. Uh, Now the ARRIVE checklist, study design according to ARRIVE checklist. For each experiment, it should give brief detail of the study design that should include: first is the number of experimental and control groups. This point was followed and the number was told. Second is any step taken to minimize the effects of objective bias when allocating animals to treatment. For example, randomization procedure, and when assessing results, for example, if done, describe who was blinded and when. This point was not followed because there was no any bias discussion was given in that, and no time limit was given. C point is the experimental unit. For example, a single animal group or cage of animal. A timeline diagram or flowchart can be used to illustrate how complex the designs were carried out. This point was also not followed because there were no flowcharts. However, tables were given, but there were no flowcharts in the methodology given. Now, ethical statement. It indicates the nature of ethical review permission, relevant license, and national institutional guidelines for the care and use of animals that cover the research. This point was followed, and as I've already told in the methods, it was being taken care of. Next is sample size according to ARRIVE checklist. First point in this should be in the list should be specify the total number of animals used in each experiment and number of animals in each experimental group. This point was followed. Next is explain how the number of animals were arrived at. This was told that animals were purchased, so the point is followed. And the next is provide details of any sample size calculation used. No details are given for the sample size calculation. So this point is also not followed. C is indicate the number of independent replication of each experiment if relevant. This point is also not told, so not followed. Next is experimental procedures according to arrive checklist. For each experiment in each experimental group, including controls, provide precise details of all procedures carried out. For example, first is how. For example, drug formulation and dose, site and route of administration, anesthesia, analgesia used, provide details of any specialist equipments used, etc. This point was followed because it was told that at the end of the experiment, halothane gas was used to kill all the rats. So this point was followed. Second is when, time of the day. This point was not followed because it is not told. Where? This point is also not told. It was told to us that they were taken in cages, but when the experiment is performed, this was not told in the article. Now D point is why. Like for example, rationale of choice of specific anesthetic, route of administration, drug use. This point is also not followed because rationale for using any drug or anesthetic was not told. Next is experimental animals according to arrive checklist. First, A is provide details of the animal used, including species, strain, sex, developmental stage, and weight. Sex was told because it was male rats. Weight was told that was about 70 gram. And name of the animals, that is rats, was shown. And remaining things, however, were not followed, that at what developmental stage they were in, and what is the median and mean for that. B point is it should further provide further relevant information such as the source of animal, international strain nomenclature, genetic modification status, for example, knockout or transgenic, genotype, health immune system, drug test, NAIF, previous procedures, etc. This point was not told at all. Next is housing and husbandry according to arrive checklist. It provides details of housing, that is type of facility, for example, specific pathogens free, type of cage or housing, bedding material, number of cage companion, tank shape or material for fish. This point was followed because it was told that they were taken into cages. B is husbandry condition, that is breeding program, light dark cycle, temperature, quality of water, type of food, access to food, environmental enrichment. This point was also followed because we were told that they were taken in light and dark 12-hour cycle each. And C point is welfare related assessment and interventions that were carried out prior to or during or after the experiment. This point was not followed. Next heading in the arrive checklist is allocating animals to experimental groups according to arrive checklist. Give full details of how animals were allocated to experimental groups, including randomization or matching if done. It was told in the article that at the age of 4 weeks, they were sorted into equal body weights and divided into 5 dietary treatment groups of 25 animals. So this point was followed. B is describe orders in which the animals in the different experimental groups were treated and assessed. This point was also followed. Introduction. Uh, the purpose of the study was to determine whether a diminished folate status might alter the development of intestinal tumors induced by azoxymethane in the male rats. A secondary objective was to test if DNA methylation was altered in the folate depleted groups. A non-absorbable antibiotic drug was employed in some dietary treatments to further enhance the level of folate depletion, both before cancer initiation and during promotional progression phase of carcinogenesis. This paragraph is added from the article and it's highlighted because these points are followed. Uh, in this experimental outcomes according to arrive checklist, that it clearly defines the primary and secondary experimental outcomes, like as told in the earlier paragraph, that primary and secondary outcomes both are given. And so this point was defined and followed. Now coming towards the statistical analysis. The DNA from colonic mucosal scraping and a small portion of the liver was isolated using a high pure template preparation kit, followed by the treatment with 10 milligram per ml RNAs, which had been preheated to 100 degrees centigrade for 15 minutes and cooled slowly to room temperature. Short chain fatty acids in the feces were measured by gas chromatography. Briefly, feces were homogenized in 3 ml of water, and 200 microliter liquid was mixed with 20 microliter of methyl and valeric acid. The mixture was then centrifuged at 10,000 gram at room temperature for 5 minutes. Of the clear supertenant was injected directly into the gas chromatograph. Group results of the biochemical markers were compared using a one-way analysis of variance followed by Turkey's multiple comparison test. Analysis of tumor incidence was carried out using chi-square test, a generalized linear model with Bayesian distribution of errors was used to analyze difference between treatments for tumor numbers in the large and small intestine. For tumor mass index, the data was logarithmically transformed to allow for normality of data and then analyzed using one-way analysis of variance, that is ANOVA. The Kruskal-Wallis test was used for ANOVA to determine group differences. Now, statistical methods according to arrived checklist. Point A is that it provides details of statistical methods used for each analysis. So, as I told in the earlier paragraphs, all these points were followed. Next is specify the unit of analysis for each database, that is single animal, group of animals, single neurons. This was not followed. Point C is that it describes any methods sorry, used to assess whether the data met the assumptions of statistical approach. No such method is followed. Now coming towards the results, I've added just two paragraphs from results. The rats in all dietary groups showed a similar weight gain. At the final week, there was no significant difference among the 5 groups. Number of tumor-bearing animals in the folate deficiency is given in the percentages. Overall, there was a significant decrease in total tumor cell, small intestinal plus colonic incidence, and number of tumors associated with the folate depleted treatments. Histopathological appraisal of colonic tumor types showed that the animals fed the folate depleted diets developed significantly fewer. Total concentration of feces and fecal pH was significantly reduced after 4 weeks as a result of incorporation at the level of 1% of the diet. And all standard deviations, mean, and for the controls and cases are given along with the pH values. These slides shows the tables that were included in the article. Now results according to arrived checklist. For baseline data, it shows that for each experimental group reports relevant characteristics and health status of animals, for example, weight, microbiological status, and drug or test name prior to treatment or testing. However, this information can often be tabulated. This point was not followed in the results. Numbers analyzed. Report the number of animals in each group included in each analysis and report absolute numbers. This point was followed because all the percentages were given in the results along with the mean and median. B point is if any animal or data were not included in an analysis, you should explain why. All animals were included in the analysis, so no such explanation is required. So this point was also not taken. Outcome and estimation of the result, that it reports the result of each analysis carried out with a measure of precision. For example, standard error or confidence interval. This point was followed. Adverse events according to results, that option A, first A is it gives details of all important adverse events in each experimental group. This point was not followed, not any information about the adverse event was given. B is that it describes any modifications to the experimental protocols made to reduce adverse events. This point was also not followed because no any adverse event was discussed. Now coming towards a paragraph of discussion before critically appraising it on checklist. The main aim of the present study was to examine the effect of folate deficiency on intestinal tumorigenesis in the rat. The results clearly demonstrate the folate deficiencies associated with the reduction in the risk of intestinal tumorigenesis induced by azoxymethane in the rats. The reduced intestinal tumor as a result of folate deficiency in the present study was not entirely unexpected, as other experimental animal studies have also been shown that folate deficiency can suppress or delay the onset of tumor. Folate plays an important role in nucleic acid biosynthesis and processes involved in cell proliferation. An association of folate supplementation with enhanced cancer induction in humans has been suggested previously, or may be an effect of requirement of tumor growth for folate. A continuation of discussion is in the present study, folate deficiency supports suppression of promotion or progression events in the intestinal cancer at both the incidence and number of intestinal tumors were reduced. However, we were unable to determine whether folate is important before development of intestinal tumor. A deficiency of folate in the later stage of tumorigenesis is certainly reduces tumor development. We thank Ben Shred and Peter Royal for technical assistance, and we would also like to acknowledge the Dairy Research and Development of Corporation for their financial support of this research. Now, discussion according to ARRIVE checklist. Interpretation or scientific implication. A is interpret the results taking into account study objectives and hypotheses, current theory, and other relevant studies in the literature. This point was followed in the discussion. Comment B is comment on the study limitation, including any potential source of bias, any limitation of the animal model, and the imprecision associated with the result. Limitation is discussed briefly, but no bias or model limits are told. C is describe any implication of your experimental method or findings for the replacement, refinement, or reduction of the use of animal in research. This point was not followed. Generalizability or translation. Comment on whether and how the findings of the study are likely to translate to other species or systems, including any relevance to human biology. This point was not followed because any discussion with the human biology was not done. Funding. List all funding sources, including grant number and the role of funders in the study. This point was followed, but the role of funders was not given in explanation. Now, strengths of the article, according to me, are beautifully written with scientific background. Abstract was given in detail. Method of the study is given in detail. Tables were given of cases and control for folate deficiency results, and sample size was appropriate. And limitations are that the title was average, objective was not given clearly, a quality appraisal tool or checklist was not provided, limitations of the study are not given, correlation with human biology is not provided. This is the YouTube link, and thank you.